These are my disclosures. VIP152 sensitivity in a panel of gynecologic cell lines has been performed. We see activity in cisplatin-sensitive and resistant cell lines with an in vitro IC50 ranging from 38 to 379 nanomolar. We use this sensitivity data coupled with baseline genomic features from DepMap to discover that tumor mutation burden, TMB, is a feature that is associated with VIP152's most sensitive quartile of cell lines. The heat map on the left shows the broad panel of gynecologic cancer cell lines, with each row being a gene mutation. Mutations are highlighted in yellow, wild type is in purple, and what you can see from the top 100 genes that the quartile of cell lines in the green box that are the most sensitive to VIP152 also harbor the most number of gene mutations and have a high TMB score. For contrast, immediately on the right, you'll see the least sensitive quartile of cell lines in the gynecologic panel. We see a similar trend of TMB predicting sensitivity to VIP152 in the ovarian subset of cell lines in the right-hand heat map. TMB association with response to VIP152 remains to be validated in an independent cohort and is planned. VIP152 cytotoxicity and MYC protein regulation have been observed in platinum-resistant or treatment-naive patient-derived ovarian and endometrial cancer tissue samples ex vivo, and you can find those on our poster. Monotherapy efficacy of VIP152 at the 17.5 mg/kg was evaluated in a cisplatin-sensitive ovarian cancer A2780 cell line xenograft model, where a single dose provides tumor growth inhibition compared with vehicle control. In an ongoing phase 1 trial of VIP152, stable disease is observed in 4 out of 7 patients with gynecologic cancers. The clinical characteristics of these patients is in the table on the slide. These patients are participating in the ongoing phase 1 VIP152 trial. And from these 7 patients, the diagnoses are 1 with fallopian tube carcinoma and 6 with ovarian cancer. All 7 patients were heavily pretreated with at least 2 prior lines of therapy. 4 out of 7 patients have achieved a best overall response of stable disease. Out of these 4 stable disease patients, 2 remain on study. 3 of these patients have known histology and biomarker profiles. They are of serous histology, microsatellite stable with MYC amplification, and a range of TMB scores from 4 to 9 mutations per megabase. Neutropenia is a non-target toxicity via the downregulation of MCL1 and is manageable. You'll see in the left panel a line plot of the 7 gynecologic cancer patients treated on the ongoing phase 1 trial, where we look at absolute neutrophil counts versus each of their visits to the clinic. VIP152 PK in these patients, estimates are within the previously reported range, both clearance and volume of distribution. In the whole blood of patients samples of 5 gynecologic cancer patients, which you see on the right-hand side, VIP152 downregulation of 3 known CDK9 target genes, MYC, MCL1, and PCNA message have been detected in all 5 patients treated and studied so far. In conclusion, VIP152 demonstrated sensitivity in gynecologic cell lines independent of cisplatin sensitivity. We show that TMB is associated with VIP152 sensitivity in cell lines of gynecologic malignancies. VIP152 induces cell toxicity as well as MYC protein reduction in platinum-resistant or treatment-naive ovarian and endometrial patient-derived cancer cells. Tumor growth inhibition by VIP152 was demonstrated in the A2780 human ovarian cancer xenograft model in vivo. In the clinic, 4 out of 7 patients with gynecologic cancers treated with VIP152 in an ongoing phase 1 trial have achieved a stable disease as their best overall response, and 2 of these 4 stable disease patients remain on treatment. Neutropenia is observed and manageable with supportive care and once-weekly dosing. VIP152 has predictable PK properties in gynecologic cancer patients, and we see gene expression modulation in all 5 patients studied. Intermittent dosing provides modulation of CDK9 regulating oncogenic targets such as MYC and MCL1. Thank you for your attention.